Let's have the hydroxyl, sulfhydryl, and disulfide groups in the side chain of your amino acids. Hydroxyl groups OH, they are usually found on the side chain of your serine, tyrosine, and tryptophan. And the hydroxyl groups can form hydrogen bonds, which means if a specific amino acid contain hydroxyl groups in its specific side chain, that side chain can basically interact with water. OH, yeah, hydroxyl group. The O here is in covalent bonding with the hydrogen. So I guess you are already familiar with this one. Right, so for example, the side chain of serine, you have CH2OH. We have threonine. As you can see, there is also a hydroxyl group here. Tyrosine, also a hydroxyl group here. So with the presence of hydroxyl groups in the side chain of the amino acids, the amino acids now can have the capability to interact with water, can form hydrogen bonding with water. Now, the first functional group that we will be reviewing on will be the sulfhydryl group. Sulfur is found mainly in proteins in the form of sulfhydryl group, which is symbolized as SH, or one sulfur atom being bonded to a hydrogen atom, or disulfide group, which is symbolized as SS, wherein there are two sulfur atoms being bonded together. Sulfur is present in cysteine, which has the SH or sulfhydryl group in its side chain, and the methionine amino acid has the specific SS or disulfide group in its side chain. The sulfhydryl groups of two cysteines can form a disulfide producing cysteine. So basically, when two cysteines are reacted together, they can be able to form one new amino acid, which is your cysteine. And take note that the difference between cysteine and cysteine is that cysteine has sulfide group, has a specific sulfide group in its side chain, whereas cysteine has disulfide group in its side chain. So later in the next slide, we'll be able to know the difference between cysteine and cysteine. Alright, so we have here the molecular structure of the two amino acids. So as you can see in this part, this one would be your cysteine, and on the side chain of cysteine is your sulfhydryl group, SH. Now, if two cysteines are reacted together to form one new amino acid, one new bond will be formed between the sulfhydryl groups of two cysteines. The two hydrogen atoms will be released during the reaction, and a bond will be formed between two sulfur atoms of the two cysteines, forming now this one new group or functional group, which is the disulfide group. So next one would be the ionizable group. Ionizable groups are present on the side chains of 5 amino acids, namely aspartate, glutamate, histidine, lysine, and arginine. So when we say ionizable groups, they have the capability to carry a charge depending on what environment they are exposed to. If the environment is acidic, basic, or just neutral. So depending again on the pH or environment they are exposed to, they will be able to carry a specific charge. When charged, these ionizable groups of the amino acids can form electrostatic interactions. Adding or removing one or more electrons gives a net electric charge to a specific a specific atom. So as you can see, we have some amino acids that have positively charged side chains. First, we have the lysine, then the arginine and histidine. They are positively charged because in their side chain they have this specific amino group attached to a specific aliphatic group, right? So we have here the amino group sa side chain ni lysine, two amino groups sa side chain ni arginine, and two amino group attached sa side chain ni histidine. Now, in physiologic pH, we know that the normal behavior of your amino group is to accept proton. So that's why your amino group of lysine here is positively charged. The amino group of arginine is also positively charged, whereas the amino group of histidine carries what? Another proton here. Now let's move on to the negatively charged R groups that we can find in the side chains of your aspartate or aspartic acid, other term. Again, aspartate or aspartic acid, and glutamate or glutamic acid. Now, as you can observe in the side chain of your aspartate and glutamate, we have what we call additional carboxyl groups in their side chains. And we already knew that in physiologic pH, again, in physiologic pH, carboxyl groups usually carry negative charge because they tend to let go the protons that they have in their structure. Now let's have the amides. Amides are present on the side chains of asparagines and glutamines. Amides are derived from carboxylic acids, whereas a carboxylic acid contains the carboxyl group, and in an amide, the OH part of that group is replaced by NH2 group. So basically, the molecular formula for amides would be C double bond O NH2. Again, C double bond O NH2. So this would be basically the group that will be formed once two amino acids are reacted to each other. So again, amides are actually groups that result from the reaction between two amino acids. In the next slide, you will know how exactly amides are formed during the reaction of two amino acids. But basically, your amides is just C double bond OOH plus the NH2. The C double bond OOH here belongs to one amino acid, and the NH2 here is the amine group or amino group found on the molecular structure of the second amino acid. So once the two amino acids react together, this part of their structures will have to be attached to one another. And in the process, the C double bond OOH or carboxyl group will lose its OH. Madula ang OH niya, mabili lang si C double bond O. And then the NH2 of the second amino acid will just have to be connected to the C double bond O of the first amino acid, forming now your amide. Now we have an example of amide groups found in the side chain of a specific amino acid. Remember in the previous slide, I've mentioned that amides can be formed when two amino acids are reacted to each other. However, amides can also be found solely in the side chains of some amino acids. For example, in the side chain of asparagine and glutamine, you have their amide groups. Again, an amide is just a combination of carboxyl group plus the amino group. So you have here a part of the carboxyl group, which is C double bond O, and then your amino group here, which is NH2. And as you can see, the two functional groups are connected to one another, forming that specific amide or a new group. Same with the glutamine. So you have here the carboxyl group, C double bond O na lang, kaya nakuhaan siya sang OH. And then you have here the amino group bonded to one another in order to form one new functional group, which is the amide group. And this is quite the characteristic, group that basically is contained in the molecular structure of your asparagine and glutamine. The side chain of proline forms a ring with a nitrogen and is attached to the alpha carbon. So notice here the structure of proline, and I guess I've already mentioned this in one of the previous slides. The side chain of proline is also attached to the amino group, forming this specific ring structure. And this makes your proline quite unique from other amino acids because this is the only amino acid that basically has um, a ring form, a ring structure wherein the side chain and the amino group are involved. Proline is not an essential amino acid, which means that the human body can synthesize it. Now let's first identify, or let's first, define what is an essential amino acid. An essential amino acid is a specific amino acid that is not produced in the body, which means the body has no whatsoever capability in producing that specific amino acid, but that body needs that specific amino acid. Hence, we get this specific essential amino acid from food sources. But in this case, your proline can be synthesized by the body, or that the body has the capability to produce proline. Hence, hence the proline is not considered as an essential amino acid. Again, proline is not an essential amino acid because the body has the capability to produce proline. Later on, I will provide the essential and non-essential amino acids in the succeeding slides. Proline is unique among the 20 protein-forming amino acids in that the amine nitrogen, which is this one, is bound to not only one but two alkyl groups. So this is the first alkyl group that is attached to your amine nitrogen, and this is the second alkyl group that is attached to your amine nitrogen. And since there are what we call two alkyl groups that are attached to your amine nitrogen, it makes now the amine nitrogen as the secondary amine. So we're basically done discussing about the hydrophobic or nonpolar amino acids, which basically have nonpolar or hydrophobic side chains in their structure. As of this moment, we will have the polar uncharged side chains that we can basically see in the structure of serine, threonine, cysteine, proline, asparagine, and glutamine. Polar because these specific side chains can be able to form polar interactions with other polar molecules, such as water. In the structure, in the side chain of serine, it has the hydroxyl group, same with this in the structure of your threonine, and yeah, the presence of hydroxyl groups in their side chains make them polar molecules. Remember that the hydroxyl groups of serine and threonine can form hydrophilic interactions with water. In the side chain of cysteine, we have the sulfhydryl group, which makes cysteine polar. For proline, well, the ring structure of proline, plus the fact that there is what we call an amino group attached in the side chain of proline, makes proline quite polar. The asparagine and glutamine has, or sorry, asparagine and glutamine have amide groups in their side chain, and amides can basically form polar interactions also. So these are the 20 standard amino acids. You can also have here the complete name as well as the abbreviations of the 20 standard amino acids. Kindly please do remember the 3-letter abbreviation as well as the 1-letter abbreviation of the 20 standard amino acids, because for sure they will come out in the examination. And the 20 standard amino acids again are categorized or classified into groups depending on the type of side chains that they have. For example, we have the non-polar aliphatic R groups, right? Kindly remember the different amino acids belonging in this group. Same with the aromatic R groups, the positively charged R groups, negatively charged R groups, and the polar uncharged R groups. Anyway, I've already made a table summarizing the amino acids belonging in different groups depending on their side chains. On the nonpolar aliphatic side, yes, kindly remember these amino acids: glycine, isoleucine, valine, alanine, leucine, methionine. For the polar uncharged, we have cysteine, asparagine, threonine, glutamine, proline, and serine. Aromatic, we have tryptophan, phenylalanine, and tyrosine. For the positively charged amino acids, we have histidine, arginine, and lysine. Negatively charged, we have the glutamate and aspartate. All right, now I guess this is the formal discussion on the classification of amino acids based on polarity and their charges. Nonpolar or hydrophobic amino acids contains one amino group, one carboxyl group, and a nonpolar side chain. So I guess you already knew this one, although what we've previously discussed would be the nonpolar hydrophobic aliphatic and aromatic amino acids. What we are going to discuss as of this moment will be quite, similar or the same with what we've discussed in the previous slides. So take note that nonpolar hydrophobic amino acids, they contain one amino group, one carboxyl group, and a nonpolar side chain. For those amino acids with nonpolar amino acid side chain, we have the valine, isoleucine, methionine, proline, phenylalanine, glycine, alanine, and tryptophan. So when we say nonpolar, it could be that the side chain of the amino acid is aliphatic or aromatic. So what we have here would be amino acids that either have aliphatic side chains or aromatic side chains. So I've already provided the abbreviation. You can use that specific, sorry, a mnemonic. You can use that one, or you can, basically make a new one which suits your taste better. All right, so we have the nonpolar aliphatic R group. Next, we have polar hydrophilic amino acids. So this is basically the opposite of the nonpolar hydrophobic amino acids. Polar hydrophilic amino acids basically can form polar interactions with water or other polar molecules. They contain one amino group in their structure, one carboxyl group, and a polar side chain. They can be categorized or further categorized as neutral hydrophilic amino acid, acidic hydrophilic amino acid, or basic or alkaline hydrophilic amino acids. For the polar neutral amino acids, when we say neutral, they are uncharged. They don't basically carry charges in their side chain. We have the cysteine, asparagine, serine, threonine, glutamine, and tyrosine. You can still use this specific mnemonics if you want to, but if you would want to make a new mnemonics for this group, then just go ahead. And yes, we have what we call the structure here of amino acids that are basically polar hydrophilic. We have polar acidic amino acids. They're acidic because they basically contain what we call another carboxyl group in their side chain. And we know that carboxyl groups tend to carry negative charge in their structure. It would contain, the amino acids that are polar acidic. They contain one amino group, one carboxyl group, and a second carboxyl group as the side chain, as I've mentioned a while ago. They're acidic as at physiologic pH, which means they carry negative charge. The side chain carboxyl group bears a negative charge, making it C double bond O negative. I guess you already knew this one. Polar acidic amino acids, we only have two, so it's really very easy for you to remember them. We have the glutamic acid or glutamate, aspartic acid or aspartate. Remember that these two amino acids were mentioned in one of the previous slides, and they contain what we call ionizable side chains, right? Ionizable in a sense that their side chains can have the capability of carrying a negative charge. So, they are known as polar acidic amino acids because their side chain carry what we call, their side chains basically carry another carboxyl group. And we know that at physiologic pH, the carboxyl group would usually carry a negative charge. Alright, so just a refresher, this is the molecular structure of your aspartate and glutamate or aspartic acid or glutamic acid. Notice that in their side chain, they have what we call a secondary carboxyl group. And we know that at physiologic pH, the carboxyl groups usually carry a negative charge. So whatever happens in the first carboxyl group in their structure also happens in the second carboxyl group that they have in their side chain, because they are just the same. Carboxyl group din siya, carboxyl group man eh. So whatever the behavior of the carboxyl group in this part of the structure will also be the same behavior the second carboxyl group will do. So we're down to the polar basic amino acids. The side chain basically is positively charged. The basic amino acids, polar basic amino acids, have one amino group, one carboxyl group, and a second amino group as the side chain. And we all know that in physiologic pH, the amino groups usually accept protons, right? Ma-positively charged sila. The polar positively charged amino acids are basic at physiologic pH. The side chain amino group bears a positive charge, making it NH3 plus. So here are the polar basic amino acids, and there are just three of them. We have histidine, lysine, and arginine, and this is the mnemonics rather that I've created. And again, you can just create your own, or you can just use this one. For their structure, you have here, yeah, let's check this out. In the side chain of lysine, we have an amino group here, right? So this is basically the second amino group in the structure of lysine because we have the first amino group here. In the structure of arginine, we have amino groups in the side chain and the amino group here. For the structure of histidine, we have amino group, first amino group, and then we have two amino groups. Now, please do remember that whatever happens in this part of the molecule, if it would carry a positive charge or not, that will also the same behavior that this specific amino group will do. Again, kung ano matabo diri sa part sa molecule, amo man niya matabo sa amino group diri nga part sa molecule. Because they're just the same. This is an amino group, and this is also an amino group. So whatever the behavior of one amino group will be the same behavior that we will observe in the other amino group. Same man day sa arginine nga molecule or amino acid. And same man kay histidine. This one and this one will have the same behavior. Now it's time to talk about the essential amino acids. Again, they are called essential amino acids because the body cannot create them, cannot produce them. Hence, we need to eat food sources in order to get the essential amino acids from them. Alright, so just what I've mentioned a couple of times, essential amino acids are standard amino acids that we need to obtain from the diet or from food sources because our body cannot synthesize it in adequate amounts from other substances. And because we need the essential amino acids in order to form new proteins, new structures in the body, we really need to take them from the food sources or from the diet. Right, so again, I've provided the mnemonics for the essential amino acids. And this is the mnemonics that I've been using since I guess high school or first year in college. But anyway, we have here phenylalanine, valine, threonine, methionine, arginine, tryptophan, histidine, isoleucine, leucine, and lysine. Those are your essential amino acids, which means these are the amino acids that basically we need to get from the diet or from the food that we eat. And that ends our discussion on amino acids. Next video will be about the peptide bonds and a little bit introduction to proteins, how proteins are formed, and also we will discuss about the different classifications of proteins based on shape and based on their function. So thank you very much for listening, and I hope you've learned you've learned a couple of new things from this video.